So yeah, welcome everybody to today's Flash Talk. Today we have our guest Patrick Schnider, and I'm here also with our CEO Dennis Hu. We are going to be talking about the Medicinal Chemist's Guide to Solving ADMET Challenges today. And with that, I will hand it off to Dennis for some more introduction.
Yes. Thank you. And again, thank you so much for joining us, Patrick. So for the audience, I think many people may know of Patrick already and his book, or the book he edited, The Medicinal Chemist's Guide to Solving ADMET Challenges. Patrick has a really impressive background. He's been working in pharma R&D as a medicinal chemist and team leader for more than 26 years. He is an inventor and co-inventor of 7 drugs which have entered clinical development, including the marketed neurokinin-1 receptor antagonist, nitupetant, for the prevention of chemotherapy-induced nausea and vomiting. Over his 22-year tenure at Hoffmann La Roche, Patrick's contributions were recognized by the Pharma Research Olympiad Gold Medal, the Medicinal Chemistry Leo Sternbach Award, the Inventorship Recognition Award, and the Roche Managers Meeting Award of Excellence. At Rejuveron, he led the incorporation of 2 affiliates and served as founding chairman of the board of directors and CEO ad interim. Prior to joining pharma, he received his academic training as a chemist at the University of Basel, the Max Planck Institute for Kohlenforschung, and the California Institute of Technology. Patrick's service to the chemistry community includes the 2019 presidency of the Basel Chemical Society, and he is currently a member of the board of the chemistry alumni of the University of Basel. Beyond drug discovery and development, Patrick has been deeply involved in working groups establishing and implementing global ADMET profiling strategies. And he is a member of the team that conceived, developed, and operated the Roche Medicinal Chemistry Knowledge Database, ROC. He took a deep dive into how to use data and information to generate and capture knowledge, something that we're very passionate about here at Drug Hunter, and effectively retrieve it, which led him to edit and co-author the Medicinal Chemist's Guide to Solving ADMET Challenges. This is a really impressive book with many, many chapters. So Patrick won't be able to tell us everything that's in this book, but I think he'll pick out some selections of his favorite scientific gems for us today. So Patrick, thank you very much for joining us.
Yeah, thank you very much, Dennis, for the kind introduction, and Dennis and Dennis for inviting me to this Flash Talk. I think this is really an exciting series, and everybody for dialing in. So I joined the pharmaceutical industry at the end of the 1990s, which was a really exciting time, in my opinion, because within a few years, a lot changed in the way how we were doing small molecule drug discovery at the time. During the 1990s, quite a number of drugs actually had to be withdrawn from the market due to safety issues, in particular liver toxicity and cardiotoxicity. And also, it was not so uncommon that many drugs failed relatively early on during development due to inadequate pharmacokinetic profiles. It was also a time when chemists started to produce much more compounds than, let's say, a decade before due to high throughput parallel chemistry. So it rapidly became clear within the industry that it was necessary to implement much broader and earlier ADMET profiling than this had been done before that time. At Roche, where I had joined as a rookie, they did this in what I thought was a very nice way by implementing those interdisciplinary working groups that Dennis had mentioned in the introduction. And the tasks of these working groups included harmonizing assays across sites, but also coming up with screening strategies and figuring out how to actually best use the data that were generated in all these ADMET assays. Now, if we look at the knowledge pyramid, we can see that data is actually at the bottom of this pyramid. Data is just the facts, and kind of not that useful per se. In order to make it more useful and turn it into information, you need to process and structure it. Now, what we ultimately want, of course, is knowledge. And for that, knowledge is condensed, essential, and actionable information. And ultimately, we want to achieve the stage of wisdom, which is difficult, wisdom being a deep understanding of knowledge, which would eventually lead to deep insights. Now, we're talking about a time 25 years ago. There were already from the beginning, when the new corporate database collecting all the data globally from all the sites was implemented to automate the step of knowledge generation from data and information. But it turned out to be more complicated than we all originally thought. And I think there are many reasons for that. But to some degree, this is intrinsic to the multifactorial nature of the data we're dealing with in ADMET, which I would like to illustrate here. For instance, with this example from a group from AstraZeneca around Andrew Leach, who had really pioneered matched molecular pair analysis. In 2006, they published that replacing an aromatic hydrogen atom by a methyl sulfonyl group would usually be expected to lead to a significant improvement in solubility. And of course, this is quite plausible because this replacement is usually leading to a significant decrease in lipophilicity by almost an order of magnitude. But 5 years later, they published that with the addition of new examples to the database, the effect had completely disappeared. And the replacement of hydrogen by methyl sulfonyl would, have no impact on solubility. And that's, of course, would be, you certainly couldn't phrase it like that, for sure. Now, this is, after all, not so surprising because solubility is a function of lipophilicity and of crystallinity. A year later, another group from AstraZeneca actually published their data on a series of aryl methylsulfone derivatives, which they found to have exceptionally low solubility. And using X-ray crystallography, they could show that one of the main reasons for this low solubility was in fact high crystallinity due to extensive hydrogen bonding between the methyl sulfonyl groups. And they could show that replacing this aryl methylsulfone group by a substituent which could not form such hydrogen bonding networks, like a pyridine, would significantly improve solubility, although the lipophilicity even went slightly up. Now, lipophilicity is clearly if not the key ADMET property. And I think this is really great news for us as chemists, because this is a property we can modify so well by structural modifications. What's important, though, is to keep in mind the interrelationship between log D, log P, and ionization. And as a quick reminder, I'd just like to give the equations showing this interrelationship for the simplest case, monobasic or monoacidic compounds. And to just illustrate this, if you have a compound with a pKa, a basic pKa of 9.4, which will be essentially completely protonated at pH 7.4, the intrinsic, excuse me, log D, which represents the effective lipophilicity, will be 2 orders of magnitude lower than the intrinsic lipophilicity expressed as log P. Whereas a compound with a pKa of 7.4, which will be 50% protonated at pH 7.4, the effective lipophilicity will be only 2-fold, or 0.3 log units lower than the intrinsic lipophilicity. Now, it is clear that that there are better and better computational methods to predict log D, log P, and also pKa values. But many of these methods, if they're not ab initio, will rely on a database on which they were trained. And I would still argue that in many cases, the outcome of such predictions will very much then depend on the diversity of data and also the amount of data that's in the database that was used to train the model or the tool. And to give one example on this, we designed a series of conformationally locked piperidines and synthesized derivatives with a total of 14 different substituents in the beta position to the basic nitrogen, which are then either locked in the equatorial or in the axial position. And so I'm just giving you here a snapshot of this with these carboxy substituents. As you can see, these are the black bars. The effect of a methyl ester or primary, secondary, tertiary amide in the equatorial position on pKa is almost the same. pKa goes down by about 1.6 units. For the carboxylate, which has a higher electron density and hence a lower electron withdrawing capacity, the effect is less pronounced. Now, for the axial derivatives, the situation is quite different. While for the ester, this is very similar as the equatorial case. The pKa reducing effect of the axial primary and secondary amide is much less pronounced. And it's actually inverted. So the pKa of this axial tertiary amide substituted derivative is in fact higher than that of the unsubstituted piperidine, and the effect is much stronger with the carboxylate. Now, how can this be explained? Amides are very good hydrogen bond acceptors. And in the case of the secondary and primary amides, you have these two conformations which will be in equilibrium. Conformation B, where the protonated state of the amine will be stabilized. This effect is actually reducing the pKa lowering effect of the carboxy substituent, while the conformation A is stabilizing the non-protonated form. Now, an ester is a much less strong H-bond acceptor than an amide, and that's the reason why this effect is much weaker for the axially substituted ester derivative. In the case of the tertiary amide, the conformation A is not possible, and that's how you could explain why this compound is ultimately actually more basic. Now, what's the impact on ionization for the equatorial derivatives? The pKa lowering effect is quite strong, and hence the degree of protonation is less than 40% for all the neutral carboxy substituents shown here. For the axial substituents, the derivatives with the axial substituents is then gradually from 50% to almost complete. Effect of these pKa differences on lipophilicity. Now, on the left-hand side, you see log P values, which were calculated from the experimental log D and experimental pKa values. And on the right-hand side, the log D values. What I find personally quite interesting and potentially useful is if you focus your attention to the amide substituents on their log D values on the right-hand side, where you can see that although the pKa was lowered significantly, and thus also the degree of deprotonation, this hardly affects their log-D, their effective lipophilicity. It actually stays about the same. The exception is the derivative with the axial tertiary amide substituent, which is really almost fully protonated and therefore has a log-D, which is reduced by about an order of magnitude. And now reducing pKa while not inflating log-D is something that we often try to do in medicinal chemistry, for instance, to mitigate hERG inhibition. And it would be really interesting to see a series of compounds and their effect on hERG, whether this has a beneficial effect. Again, the ester substituent, which is of course a bit less useful in medicinal chemistry anyway, due to its hydrolytic liability, in fact increases the effective lipophilicity log-T, although it has a slightly log-P lowering effect. Now, there was another case, this vasopressin 1A receptor antagonist program, where log-D and hydrogen bond acceptor basicity played a role. On the right-hand side, you see the logarithmic PGP efflux ratios plotted against log-D. And it looks like there is a trend so that you could say log-D may be useful in guiding your design and your optimization. But the correlation on the left-hand side seems to look rather better, although this is really terribly simplified, I should say, because we used just literature values for the R groups and just the values for the strongest H-bond acceptors in these R groups. Several of the R groups, in fact, have multiple H-bond acceptors, in particular the ones with higher efflux ratios. Now, if you look at some structures, the pyridine substituent is actually quite a good H-bond acceptor, and therefore PGP efflux is a bit on the high end, while the pyrazine nitrogens both have a significantly lower H-bond basicity because they mutually withdraw electrons from each other and thus lower their H-bond basicity. And therefore, we also observe a reduced PGP efflux. Pyrimidine is kind of similar in terms of H-bond basicity as pyrazine, a bit higher, and pyridazine is even a little higher than pyridine. And this compound is also really, really polar. We we found that you can indeed effectively reduce H-bond basicity by substituting the heterocycles with electron withdrawing groups, such as halogens, or an oxygen. So squeezing an oxygen into the heterocycle and the cyclohexyl ring produced, for instance, these two compounds, the pyridoxy and the pyrazinoxy group, where we essentially abolished PGP-mediated efflux along with a further reduction of the hydrogen bonding basicity. I think it is noteworthy that in terms of H-bond basicity, this pyrazine ether is actually quite similar to a phenyl ether, but it is more than an order of magnitude more polar than the phenyl ether. Another group that's quite interesting, I think, is this oxadiazole ring, where again, the three heteroatoms actually withdraw an electron density from each other, thus effectively reducing H-bond basicity. Now, prior to this work, I'm aware of one publication from Pfizer from 2008, where H-bond basicities of 5 derivatives were associated with PGP-mediated efflux. But otherwise, it's clear we know that H-bonding plays a very important role for PGP. In particular, a particularly strong effect have H-bond donors, and it is clear that overall basicity is also important, but up to this point, you would say, okay, this is an interesting piece of information. Researchers from Merck read our publication, picked up this information, and wrote it into their internal medicinal chemistry knowledge sharing tool. And so apparently it generated some interest, and people retrospectively analyzed their CNS programs and also started to prospectively use hydrogen bond basicities. And in fact, write that they found that these hydrogen bond basicities may indeed be quite useful in understanding PGP efflux. And so I think this is actually a kind of a nice example showing how knowledge can in fact be generated and captured through relatively established tools such as publications, books, and also text-based internal databases, if you like. Now, since we had difficulties coming up with artificial intelligence type models early on at Roche, we had also built such an internal Roche medicinal chemistry knowledge application called ROC in 2005. And you can, of course, search this database. It's substructure searchable, but also searchable by text, full text, or also by categories, since there are metadata with each entry into the database. But you can also browse the database, and what you see when you browse it is this kind of journal type table of content with a graphical part and a really very concise abstract that should tell the reader quickly whether the finding in this entry might be relevant for the reader to solve his own the problems in his own project or not. And if you click on the PowerPoint hyperlink, you get to PowerPoint slides where the authors who submitted the entry could elaborate in more detail on what their findings were. Now, this database, and in particular also this talk abstract, was certainly an important source of inspiration for the Medicinal Chemist's Guide to Solving ADMET Challenges. Another important source was Wikipedia. For me, Wikipedia is really a great tool because it really allows the reader to get up to speed very rapidly. You don't need to spend a long time reading long articles. And in addition, you also get key references in case you want to dig more into the details. But we should also keep in mind and remember that in organic chemistry, there is a great tradition of reference books that had this Wikipedia-type spirit already long before Wikipedia was created. And I'd like to mention in particular Green's protective groups in organic synthesis and Jerry March's advanced organic chemistry. And they're out now in the 6th and in the 8th edition, and I think this really gives testament of their usefulness and popularity by the user. And I think many chemists will have these books at their fingertips on their desks. And the idea was like, why don't we try to create something similar for optimizing ADMET challenges. Of course, I should at this point also mention the Handbook of Medicinal Chemistry, also by the Royal Society of Chemistry publisher, which is really a fantastic encyclopedic resource, which covers the pretty much all aspects that are important in medicinal chemistry and drug discovery. And of course, this book also covers ADMET aspects. What I would say, we tried to design the Medicinal Chemist's Guide to Solving ADMET Challenges in a way that it complements the handbook. As already mentioned by Dennis, the book, about 500 pages, has 19 chapters, 18 of which are on ADMET properties or a group of related ADMET properties, and I think this is pretty comprehensive. It covers, I would say, really most of the ADMET properties that are commonly optimized. Yeah, the format of the chapter is relatively standardized to help the reader quickly find the information that they are looking for. So that and it consists of two major parts. The first part is an introduction that will make the reader familiar with this particular parameter or topic. Like here in the case of cytochrome P450 metabolism, it tells you about the P450 enzyme family, their expression, structure, their function, what kinds of transformations they catalyze, and what the mechanism by which they do this is. The second important part of this introduction is a very brief summary and also key references of the screening strategies. In some chapters, this will also have decision trees and where relevant regulatory aspects. And last but not least, there is also a concise abstract on the relevance of why you may need or want to optimize this particular property. This introductory part is visually offset by this box of key references, where around half a dozen key publications will cover the topic broadly, or the entirety of these half a dozen publications does. The second part then, I would say is relatively unique. It is on the actual mitigation strategies for this particular parameter. And these are really the most proven strategies, about 3 to 6 tactics. The selection of these tactics is of course based on the experience knowledge of the authors, but also very, very thorough reviews of the literature, which will then include both individual examples and studies such as statistical analyses. And in the second part of this mitigation strategy, half of the chapters, each of these mitigation strategies will be illustrated by examples. Now, again, in a highly visual table of contents graphical style, with lots of chemical structures, since, you know, with chemists, we like to see structures and we can grasp them very rapidly. And this should give you an inspiration, you know, whether a particular strategy will be useful to solve your own project challenge or not, and maybe give you some ideas where, as the abstract very briefly explained, how the particular problem was solved in that case. Now, I hope that with this talk, I was able to convince you of the value of sharing and gathering individual knowledge, and to actually create a higher level of knowledge, and also to capture it in let's say, relatively conventional ways, be this in a book like the Medicinal Chemist's Guide to Solving ADMET Challenges, or in an online based platform like Drug Hunter, where exactly this is done. It is, you know, scientists, particular medicinal chemists, who intellectually digest information and knowledge, bring it to the point, distill out the essence, and summarize it so that other medicinal chemists can very quickly get up to speed. This leaves me with thanking quite a number of people, in fact. So first, I'd like to thank Alexander Mayweg, Andrew Thomas, and Sarah Scarrett, who played very important roles in the inception of this book project. And it was actually Alexander Mayweg as my manager at the time who triggered that I start working on a concept for such a book. Of course, the Royal Society of Chemistry book team, and importantly, a really fantastic team of authors composed of very experienced medicinal chemists and ADMET subject experts, a group that was really very dedicated and fully bought into the concept of this book. And again, of course, I'd like to thank you for listening in.
All right, thank you very much, Patrick. That is an incredibly impressive list of co-authors. This looks like a very useful resource for the community. I'm sure right after this talk, you'll see many orders for this book, for this group. So for the audience, Patrick has very graciously agreed to stay on a little bit longer to answer questions. We can sort of treat this as a free ADMET consulting session. So if you have a favorite, or maybe not favorite, most concerning ADMET challenge that you're dealing with, you can put that in the Q&A box, and we'll see if Patrick can try to help answer some of those questions live. While people are thinking of their questions to put in the Q&A box, Patrick, I have a question of my own, and also just to make sure I understood what I view as 4 key lessons from the first part of your talk. It sounds like lipophilicity is key, obviously. Chemical conformation can have surprising impacts, order of magnitude impact on pKa, log D, and solubility. The pKHB can be a better surrogate for efflux than log D. The magnitude of that was quite surprising. And positively charged compounds should be maybe carefully watched for selectivity issues. Would you say those are 4 of the key points?
Yeah, from the examples presented. I mean, overall, I think just to make this point again, and those people who know me will maybe at least smile a little bit. I think log D is really so important. Of course, you can ignore it, but I also extensively analyzed the in vitro ADMET data, all the in vitro ADMET data available at the time, back at my time at Roche. And in most cases, it was pretty clear how important log D in fact was. It is not equally important everywhere. In some cases, of course, increasing lipophilicity may be somewhat beneficial, of course, but most people know is for permeability, although you should also be careful there. There is in fact a bell-shaped relationship between permeability and lipophilicity. If you go to lipophilicities which are too high, the solubilities of your compounds tend to drop really too much, and the consequence is that this will also reduce permeability, because the compounds will, thanks to the high lipophilicity, partition into the membrane, but cannot really get out of the membrane again.
Yeah, makes sense. Absolutely. All right, so I think we can start taking audience questions. So a question from Antran is, for charged molecules, is it recommended to look at both log P and log D at the same time? Or in certain contexts, is log P a more important parameter, or vice versa?
Yeah, this is a great question. And I think the analysis that I conducted suggests that in most cases, log D is probably the better property to watch when optimizing your compounds. However, for some of the ADMET properties, at least based on the Roche data that were available at the time, the situation was a little less clear whether log P might actually be a better predictor. And the difficulty, I think, is that when people are talking of log P in publications, it's predominantly calculated log P values. And those are really, in some cases, dramatically off. So what we would actually need is more examples of really curated data sets where you have experimental log D and experimental pKa values to calculate the true log P values. Now, the difficulty with that is that with compounds with multiple ionizable groups, this can actually quickly get quite challenging. But yeah, so the jury is still out on that, and if people have nice data sets that maybe shed a little more light on these questions, on this question, I would really encourage them to publish the data, and then again, eventually, maybe we can really generate knowledge from that.
There's also a follow-up question maybe on this, Patrick, about like a software tool that you would recommend maybe to predict log D and log P. Is there any, like, I mean, of course, it's important to have a complete data set first, but is there anything that you had found successful in the past?
Yeah, I mean, there are a number of commercial tools that are quite good. I don't really want to annoy any of the providers that I might forget to mention. Ab initio would tend to give you the best results. I've seen really very impressive calculations there, but of course this is extremely computational resource intensive and not really feasible for larger data sets. At Roche, what was really useful, thanks to a good quality and quite sizable internal data set for many ADMET properties, there were machine learning models that worked really well. But there, it was also clear that when you came up with a totally different and unprecedented chemotype, the model first needed to be trained in order to provide reliable predictions.
And, maybe in another type of strategy, somebody, Jacek in the audience, is wondering about your favorite strategies to address time-dependent inhibition. So it seems like they have strong metabolism-dependent CYP inhibition. So is there, like, from your experience, what would be the kind of things you would do first in this kinds of instance?
Yeah, that's of course where we're starting to dive into the book. Of course, a lot of the reactive metabolite issues come from the fact that the compounds are metabolized at all. So again, lowering lipophilicity to reduce the rate of metabolism can in some cases help. Then there are, of course, certain structural features that would lead to reactive intermediates, such as Michael-type acceptors, quinones, for instance, or nitrines should be avoided. So you would want to replace those moieties. And yeah, for further reading, I'd like to refer you to either to the book or also to the literature. And Amit Kolgatkar, the author of the chapter, really published a lot on this.
Yeah. Sure, Dennis, you've probably experienced this as well. I think when I've seen this before, MetID has been phenomenally helpful for finding things. In one case, we were really surprised. It was a aryl methyl group that was metabolized to an aldehyde, and the aldehyde was a covalent inhibitor of the CYP enzyme, which was interesting.
Blocking those sites of metabolism.
And blocking the site of metabolism can sometimes be the solution, but sometimes it actually makes things worse. So you should be really careful.
Absolutely.
And then the tricky thing is that even relatively low levels of a reactive metabolite can cause quite a lot of problems, in particular if the drug is given in relatively high doses, and if you start to saturate metabolism, for instance, due to drug-drug interactions also. So this topic quite quickly becomes pretty complex. I think one of the goals we should generally have as medicinal chemists is when we design compounds, not just to try to avoid metabolism, but also to design the molecules in such a way that they will be metabolized in a benign way, that is, produce metabolites that will be excreted cleanly.
Yes. So on a related note, there's another question about what would be the best way to reduce CYP inhibition? So, rather than time-dependent inhibition, just CYP inhibition in general, CYP3A4.
Yeah. So again, very nicely covered in the book. Of course, there are many isoforms. The book covers the 6 most important isoforms. And also, what they tend to have in common is that the iron of the heme, the cytochrome, can be coordinated by groups such as nitrogen-containing heterocycles. And while we really love these heterocycles in medicinal chemistry because they're a really good way to bring in or to reduce lipophilicity generally, sometimes they really cause awful problems. So sometimes you may need to replace them, but sometimes it's already sufficient to either block the position in alpha to the nitrogen, which is likely coordinating the heme iron, and then the methyl group is often already sufficient, or again to reduce overall lipophilicity further.
Yes. Thank you, Patrick. I think given how many questions there are, we'll try to prioritize questions from non-anonymous attendees. So I see Yoshi Ishihara has a great question on what are your thoughts on percent unbound in plasma protein binding influencing ADMET? Yoshi has seen cases of percent unbound PPB increasing or decreasing or both improving ADMET or worsening ADMET. Concepts like solubility and permeability are quite straightforward to him. Increasing often equals ADME improvement, but percent unbound in PPB feels like it can swing ADME either way.
Yeah, another great and also big question. So there is no dedicated chapter to plasma protein binding or to volume of distribution, although these are, of course, 2 properties that we can also address very, very well as chemists by structural changes. So both in both cases, again, lipophilicity plays a role. The higher the lipophilicity, the higher plasma protein binding, and then volume of distribution. In the case of volume of distribution, it is primarily basic amphiphilic or basic lipophilic amphiphilic compounds that have very high volumes of distribution, whereas acids, which are largely deprotonated under physiological conditions, will have low volumes of distribution. And for plasma protein binding, there is really a very good correlation, again, for basic and for neutral compounds with measured log D. whereas there isn't really a correlation for acids. Acids generally tend to be relatively high plasma protein binding. Now, yes, absolutely, like also with regard to your pharmacology, plasma protein binding is absolutely essential. Also in predicting your efficacious target concentrations. You absolutely need to take this into account. And this is something I generally really recommend to do as early as you can. But the point is that you can't really give a general recipe, you know, like a rule type recipe, what values of protein binding you should target. Because high protein binding or volume of distribution can in fact be a good thing, because it can help you extend the half-life of your molecule. So you can't say that you should just systematically minimize plasma protein binding. On the other hand, when plasma protein binding is extensive, below what's measurable with the usual assays that we have at our disposition, then of course you have to be aware that your calculations, that is, those predictions, will get really inaccurate. And for this reason, you know, I would still try to address this. If you see that your plasma protein binding goes below about 1%, you may want to look if you could actually bring this up a little bit, but it's absolutely not essential for a drug to be successful. There are many drugs that have higher plasma protein binding, which are very good and which are on the market.
No, that was a very nice answer. Thank you, Patrick. We have another question here from Mahamuda, who is talking or is wondering about GI selectivity. So in cases where you want the compound kind of not really being absorbed, is there any advice that you would have for how maybe to design a compound with more GI selectivity?
Yeah, you minimize permeability. Yeah.
Thank you,
which is often actually easier than to bring it.
Yeah, I think one of the things that I can kind of briefly comment on, and one of the interesting things that we actually used at Novartis back then was the engineering of a soft drug. So basically something that, you know, is being absorbed and then basically in the plasma decomposed. So, you know, something can stay in the gut, but being absorbed a little bit and then in the plasma being decomposed. So just one strategy we've used on an interesting program.
Yeah, there are prodrugs, but for GI selectivity, I'm not sure if you would choose prodrug approaches. But yeah, you make it very polar, for instance.
Yeah. I think a good rule.
The difficulty can there be, of course, that the pH changes through the GI tract, where it's very acidic in the stomach. It gets more and more basic further down in your intestine. Yeah, you when designing so the ionization state of your compound, you have to keep this in mind, because when this changes, you may suddenly actually get absorption in the
Probably a good rule or law for medicinal chemistry is if a property is desired, it will be more difficult than anticipated to optimize for.
Yeah.
Okay, a question from, let's see here. So Simon Mugisa asks about CYP inhibition again, which we did answer, and then cardiovascular side effects like QT prolongation. I know next week we'll be doing a dedicated session on hERG, but Patrick, do you have any thoughts on QT prolongation in general?
Yeah, QT prolongation is of course attributed mainly to hERG inhibition. one thing that I'd also like to point out here is that you should not only look at hERG inhibition, but also at inhibition of sodium and calcium channels. And again, in the book, the chapter is called cardiac ion channel inhibition or mitigating cardiac ion channel inhibition. So it also covers NaV 1.5 and CaV 1.2 channels. And this is important because there are drugs which are in fact pan cardiac ion channel inhibitors. They kind of moderately inhibit all of those channels, and the consequence of this is that the inhibitory effects on all these channels cancels each other out, so that you actually don't see QT prolongation. So be cautious, when you really have an issue with cardiac ion channel inhibition. We also, in fact, had a project where we noticed this relatively late, where the compounds actually turned out to be sodium and calcium channel inhibitors, which also causes problems. AV block is the clinical manifestation of that, while hERG wasn't really inhibited.
Interesting.
Yeah. An interesting question about metabolism here. So for in vitro metabolism, Timothy is wondering of what type of assay you would prefer to use. Would it be like a human hepatocyte assay or an S9 assay? And then could the hepatocyte assay detect anything that the S9 fraction wouldn't be able to detect?
Yeah, they of course give different answers, right? So the hepatocytes are generally considered the gold standard. In particular, human hepatocytes are also much more expensive, so they tend to be used less, even for large companies. This is in fact a topic. Now the hepatocytes have the advantage, if you like, that they also reflect the cellular permeability, at least of the permeability of the compound into the hepatocytes, where then metabolism takes place. When you use non-cellular system, of course, you lose that part. Which could then, of course, lead to overprediction of the true rate of metabolism. Whereas, of course, when a compound doesn't enter hepatocytes very well, then there may also be reduced hepatic metabolism or oxidative CYP-mediated metabolism in particular. But again, of course, these when you have such cases, it could quickly get quite difficult. You need to go to in vivo relatively early on and see if you understand how your compound is metabolized and ultimately excreted. Otherwise, you could be seriously misguided.
Yeah. Thank you, Patrick. Actually, on that topic, I'm curious if you've ever seen any examples of so I remember on a project, and I still don't know if there are good published examples of this. I'm sure you would know. The hepatocytes, the whole hepatocytes, they express transporters, both uptake and efflux transporters. And I've seen effects where S9 fraction, you see very different trends from the hepatocytes because the molecules are either being, prevented from entering the hepatocytes or being taken up by the hepatocytes to be metabolized more or less quickly. So is that something that people have published on?
Yeah, not so much actually. Of course, we also need to look at the relative expression levels of the various transporters there. Yeah. this I think this is about as much as I'd like to say
Sure, yeah,
because
yeah.
this is now really becoming very much expert territory. But yeah, indeed, spontaneously, I'm not really aware of publications that that really discuss this in detail.
Maybe one question, Patrick. We talked a little or a lot actually about individual strategies to modulate very specific properties here. I'd be really curious to hear your perspective on, holistic drug discovery in general. Whether just a few words on how your drug discovery philosophy is. When is a compound good
Yes.
enough? That'd
Yeah.
be really
No,
awesome.
absolutely. A great question. Thank you for that one. No, this is really very close to my heart indeed. You could criticize what's the point with this book, chapters by individual ADMET properties. Of course, it is simplistic to optimize just for a single property. And as I gave an example. If you only look at hERG for your cardiac safety, this is clearly going to be overly simplistic. Now, what I believe is really important is that you, as mentioned also early on, try to do dose predictions. You need to understand what concentration of your drug you need to reach for how long in your target tissue, and analyze your compounds by whether they have the right properties to do that. Try to come up with initial predictions, and this will help you understand which parameters you should actually optimize first. And I would always do this first, and only if this starts to be in kind of a reasonable range of a predicted efficacious dose, I would start to optimize for more safety related aspects. That said, of course, as chemists, we always keep the safety aspects at the back of our heads, right? So if you build in a structure where you know that upon metabolism you could easily form a reactive metabolite, be it anilines or precursors of quinones, again, you may want to test early on. But there are also drugs with such structural features on the market, which are perfectly safe, also furans, thiophenes, and so on. If you look at those compounds carefully, you find out they're so polar that they actually don't get metabolized by CYP or in the liver, right? And so these features are not an issue because these reactive metabolites are never formed. But I think everybody who has practiced medicinal chemistry knows that at the end of the day, optimizing for particular parameters is inevitable. You'll be confronted with it, and I believe that for this reason, the approach that we chose for this book to give a guidance on what the most proven, most promising strategies that you should try first is quite useful.
All right. Well, thank you, Patrick. The time went by incredibly quickly, so it was very helpful 30 minutes. Thank you for staying for extra questions. I do see some very interesting other questions that we didn't have time to get to, so maybe, Dennis, we will try to coordinate answering these online on Drug Hunter. Mm-hmm. But again, thank you so much for taking the time late in the day to stay with us, Patrick.
All right.
Thank you
for that as well. So thank you, Patrick, for coming today and for giving an incredible presentation and staying here as our personal ADMET consultant. Very, very much appreciated.
Absolutely. And we'll see you in Basel next month.
All right. Take care.